Dear students, today's class on the topic nucleic acids. We know that DNA and RNA are the nucleic acids. Here is the deoxyribose nucleic acid and ribose nucleic acid. Nucleic acids are the heteropolymers, means different kind of polymers of nucleotides. Nucleotides. Okay, it has a many nucleotides are linked to form a polynucleotide. Polynucleotide means what, many nucleotides arranged. Here, what is a nucleotide? Nucleotide contain one sugar molecule, nitrogen base, and phosphate group. Nucleotide, TIDE, tide. It means you can remember that it has a three components. One is the sugar component, other one is the nitrogen base, other one is the phosphate group. Nitrogen bases, you know that purines and pyrimidines. Sugar is the ribose sugar, right? Phosphate group. So these all together called as nucleotide. Got it? Nucleotide have a C. Nucleoside means nitrogen base plus sugar only. Phosphate, if you add, that is a nucleotide, right? If these two, if it is there, we call it nucleoside. Along with the two, have to add phosphate, POOH. This we call as a nucleotide, right? Next, yeah, nucleic acids are the two kinds, which are the DNA and RNA. What are the two kinds? DNA and RNA. Right? DNA double helical structure is there. RNA have a single strand. These two are the genetic materials, but DNA is the major genetic material in the eukaryotes, and RNA, some virus, example, HIV virus have a RNA virus. Right? Here is the secondary structure of DNA. It was discovered by the Watson and Crick. This is the double helix model, right? See here how it is double helix model there. We learn in the 12th molecular basis of inheritance. There are more than dozen forms say DNA such as the A, B, C, D, E, Z, etc. Okay, there are more than a dozen forms of DNA. Different forms are there: A type, B type, C type, D type, E type, and Z type. DNA consists of two polynucleotide strands. How many? This is the one polynucleotide strand arranged anti-parallel to as a double helix. Anti-polar means what? No, this is the 5 prime, ends with the 3 prime, and 3 start here, ends with the 5. Okay, this is called as an anti-parallelly arranged polynucleotide strand. We called as a double helical strand, right? See here, they have given as a in DNA nucleotide consists of nucleotide. What I told, T means 3. What are the 3 component? Nitrogen base, deoxyribose, and phosphate group. These 3 together called as a nucleotide. Nucleoside means what? No, just except phosphate. Phosphate won't be there. That's known as the nucleoside. Here is the phosphate group is there. Pentose sugar is there. These are the purines and pyrimidines, nitrogen bases. Right? With that it will be made. Now backbone of the DNA is formed by the sugar phosphate and sugar chain. Backbone of the other strand backbone is sugar phosphate, and steps are formed by the nitrogen base pairs. These steps there, no, like a ladder steps, nitrogen bases. Nitrogen bases includes the adenine, guanine, thymine, cytosine, and uracil. How can you remember these as a which are the purines, which are the pyrimidines? Right? Here is a C. Purines are the adenine and guanine are the purines, AG, and CUT are the pyrimidines. Now adenine pairs with the thymine, complementary base what? No, A double bond T by the 2 hydrogen bonds. But guanine pairs with the cytosine with the triple bond hydrogen bonds. Okay, A and Z are the purines, CUT, cytosine, uracil, thymine are the pyrimidines, right? But complementary base pair are the A is equal to means what? Double bond to G triple bond C, right? A phosphate molecule linked with the third carbon of the sugar of the nucleotide at the 5 carbon of the sugar of the next nucleotide. This is very important. Phosphate molecule links with the third carbon. See, phosphate links with the third carbon and the nucleotide to the fifth carbon of the sugar of the next nucleotide. Right? That arrangement is that like that. See, between these two have a ester bond between the phosphate and OH of sugar. This is the phosphate and OH sugar. The bond is called as ester bond. As there is one such ester bond on either side called as a phosphodiester bond. How many? Two. If it is there not two ends, no, that is the phosphodiester bond. The bond between the sugar and nitrogen base pair called the N-glycosidic bond. See, this is the sugar and this one is the nitrogen base. This between this bond is the N-glycosidic bond. N-glycosidic bond present between the sugar and nitrogen base. Sugar and nitrogen base, right? That's a very important one. In a B bond, B DNA, one full turn. See here, 10 steps. 10 base pairs there. 1, 2, 3, like that there. No, 10, one full turn have a 10 base pairs are present in the B DNA. Length of one full turn length, if you calculate, that is the 34 angstroms. If you each step, how much? No, 3.4, right? 3.4 into 10, it comes as 34, right? At each step, the strand turn 30-60 degree or the 360 for full turn, right? At each step, the strand turns 36. At a length, they have given a full turn 10 base pairs. Length of the full turn 34. It is the angstrom 33.4, and that each strand turns the 30 degree centigrade, right? 30 degree, right? Right? That's the complete about the nucleic acid. We'll take one more lecture after the next video. Thank you.